Hi, everyone. My name is Karen Zeminski, and welcome back to the Human Body Systems training video. This time, I'm going to lump together lessons 5, 6, and 7 all in one video because these are a bit more straightforward than the ones in the past. First of all, I want to start by explaining that you're going to explore digestion in the stomach in lesson 5. And in doing so, students are going to learn that there are 3 layers of muscles that churn the food, as well as how the gastric juices, what they're composed of, and how they work. Now, as you're doing this is a 2-day experiment. So on your lab sheet, I find it easiest to set up the subtitles on the chart for my students to help keep their data organized for day 1 and for day 2. Now, prior to this, you're going to have to also introduce volume and how to calculate volume. Students are going to see that both mechanical and chemical digestion happen once they start analyzing what the chemicals they're using in the stomach are. And they do this by filling test tubes with gastric juice, with pepsin, and also with hydrochloric acid. Now, in order for this to yield the perfect results, you're going to have to do some doctoring of the results before the students enter if you really want that aha moment. Because I teach a multilingual class and 2 inclusion classes, I also found it very important to include a ton of visuals. I developed these using the background information that's right in the teacher's guide, and it helps me with my day-to-day lessons as I'm setting up for the experiment. Volume is also a skill that's going to need to be taught prior to this lab. I pre-teach it as a vocabulary word first and then teach them how to do the calculations. Students are going to need to determine the starting volume of their egg white, which they're going to place 3 egg whites in 3 test tubes with 3 separate chemicals. So they're going to start by figuring out the starting volume of the egg white, and then the following day, they're going to calculate the volume again to determine if there was a chemical breakdown which decreased the volume of the egg. In order to ensure that your results are accurate, you may need to go back to the test tube that has the gastric juice, take the egg out of that test tube, and I use a small scissor and just cut a piece off of every egg in a gastric juice test tube. I do this little bit of cheating because I want my students to have that aha moment that that's the one that broke down. Otherwise, they tend to get caught up on the math because the breakdown is, in fact, so small because we're using chemicals that are not as strong, and we're also not doing it for the proper amount of time sometimes. And there are so many different things working against us that I find doing this for my students lets them really kind of have that aha moment. And to me, that's more meaningful than having the exact mathematical calculation down to the thousandth place. So it's up to you how you want to proceed with it. There will be a breakdown. It just won't be as big. Introducing the measurements and telling students how to find the meniscus are also important. This picture is taken right from the teacher's guide. It's also in the student book for kids to see. Now, after finishing lesson 5, move on to lesson 6. And this is where they're going to start talking about the concept of diffusion. And that's the main goal. Diffusion and absorption is the main goal for lessons 6 and 7. And we want students to understand basically that when food is broken down, it diffuses into our cells, and that's how it provides our bodies the nutrients they need to sustain life. There's a great segue activity that uses balloons to explain the process of diffusion. And you'll fill it up with anise, and the smell will fill the classroom. Be careful of students who have asthma. They might not want to breathe too close to it. It tends to irritate some of the students, especially those with severe allergies and things like that. But it's an excellent, short, quick activity, and it allows the students to actually experience diffusion for themselves. I then ask them to do some reflecting on diffusion so that we can then connect it to the illustration. I am a frustrated artist and I draw lots of pictures. Students will put them in their notebooks and write little notes on them to themselves to help trigger memories. Now, during lesson 6, the kids are going to be using a dialysis membrane to determine which nutrients, either sugar or starch, can diffuse through the membrane. It's also important that you keep the membrane soaking in a beaker of water until they're ready to be used. This is what the dialysis membrane looks like. When you get it wet, it's the easiest way to open one end of it by smushing it between your fingers back and forth, left to right. You'll get the end to open so that you can fill it. You also want to make sure that the kids are correctly tying the bottom of the membrane so that nothing leaks out from it. By the end of lesson 6, students are better able to understand the diffusion of chemicals and various nutrients in the duodenum section of the small intestine. And part of that process involves liver being stored in the gallbladder, bile being made in the liver, stored in the gallbladder, and pumped into the duodenum. They start to understand the breakdown of fat. Now, all of these lessons connect back to the nutrients activity, where they got all of this information but didn't really know how to use it, as well as in their Pepe and Bolo reading. So in addition to your lessons, your mini lessons, and then the bigger investigations and the analyzation of the investigations, overall, the students are still having additional reinforcement through the reading, and the readings are excellent. Now, lesson 7 starts to talk about surface area. In order to determine how we digest our food so quickly, students are going to need to understand what the interior surface of the small intestine looks like. This is first determined by completing a ticket activity that will allow students to visualize that the interior surface of the small intestine is not smooth. Instead, it has all these different folds. Now, this year, I created a worksheet to help my multilingual students stay on task and walk them through the calculations. I found it to be fairly helpful. Because of the complexity of these topics, there are additional readings in the student guide that help to further explain the concepts. For example, the book will explain diffusion and particle movement by using a diagram of a cup of hot tea and what happens when you put the tea bag in. There are lots of different ways to demonstrate this. The tea bag one tends to be less material heavy, and it's a nice way for the kids to see it right in front of their own eyes. Now Pepe and Bolo have reached the end of their journey in the digestive system. Students by now should understand that digestion is our body's ability to break down the food we eat into a form our body needs to sustain life. These nutrients then diffuse into our bloodstream to ensure our overall health. And as you can see, Pepe and Bolo have barely escaped the toilet bowl, and the next time we see them will be in the respiratory system. Thanks for watching.